Hi, I'm Katharina Schmid of the Katharina T lab of the LMU Munich, and here I want to show you the results of our benchmarking where we look into copy number variant calling from scRNA-seq data. Copy number variation describes the gain or loss of genomic regions. This can be small segments up to full chromosomes. CNVs play an important role, especially in cancer, where different cancer types show different hallmark CNVs. Over time, tumor cells can evolve into different subclones with different CNV profiles. These different profiles influence the cancer progression and treatment outcome. To fully capture the heterogeneity in our cancer datasets, we can use single-cell sequencing data, from which we can also capture the per-cell CNV profile. For this, we can use different single-cell technologies, such as single-cell DNA sequencing, bisulfite sequencing, RNA-seq, or ATAC-seq. They are all based on the same principle, that the CNVs can be inferred directly from the coverage profile of the reads. We are especially interested into single-cell RNA-seq because there are a lot of datasets out there, and because of this, also many methods for CNV calling from single-cell RNA-seq have been developed. However, there exists no independent benchmarking at the moment. We are interested into two aspects. In general, how good is a CNV calling from single-cell RNA-seq compared to single-cell DNA sequencing, which would be the direct readout of the genome. And then second, of course, which method performs the best. We developed a benchmarking pipeline and looked into 6 different CNV callers for single-cell RNA-seq so far. They are all based on the same principle. If we have copy number gains in this region, the gene should have a higher expression, and the other way around, for copy number losses, there should be lower expression of the genes in this region. To get these higher and lower expression, we compare to a reference datasets of a matched diploid cell type. That this reference is required is a general disadvantage of all single-cell RNA-seq tools. We have 6 different methods in our pipeline that are based on hidden Markov models, mixture models, or segmentation approaches. All of them require the count matrix as an input, and 2 of them additionally also use allele frequencies, which they call directly from the single-cell RNA-seq reads. We evaluate the tools on 15 different test datasets from cancer cell lines, primary cancer tissue, and also diploid tissue. And for each of them, we include a ground truth from genomic data from different readouts, so either whole genome sequencing, whole exome sequencing, or in case of the diploid dataset, we assume a uniform diploid baseline. Then we compare the ground truths to the CNV predictions of each methods and evaluate the performance using different metrics. Here you can see the results of one example dataset, the SNU 601 cell line. In the top row, we have the results from our ground truths, which we take from single-cell whole genome sequencing data. This is already aggregated to pseudobulk so that we get a mean CNV profile across all cells. As the single-cell whole genome sequencing and the single-cell RNA-seq was not done exactly on the same cell, we compare the predictions on pseudobulk level. Then in the rows below, you see the results from the different RNA callers. You can see that, for example, the gain in chromosome 3 or the loss in chromosome 4 is nicely captured by all methods. But of course, we use then quantitative metrics to fully evaluate this. This is the performance across all cancer datasets. On the left, you can see the results looking into the maximal of 1 score, and on the right, looking into the Pearson correlation compared to the ground truth. Both metrics show the same, that the performance varies much more between datasets than between methods. When we explored this further, we identified several dataset characteristics to explain these differences, for example, the dataset coverage. In contrast, the deviation between methods is relatively small, and there is no clear best method. When we look into the prediction of gains and losses separately, we see that most of the methods are better at predicting gains than at predicting losses. When we look into our diploid dataset, we take CD4 T cells from a PBMC dataset, and we expect that all the methods should predict only base, so that the karyogram should be completely white. As you can see, If we use CD4 T cells as a reference again, so the exactly matching cell type, this is working very well. But we also tested other references, and if we use, for example, CD14 monocytes as a reference, suddenly we get a lot of spurious CNVs. This shows us that the selection of the best matching reference is really an important factor for the performance. Interestingly, numbered CNVs also identifies the diploid baseline when we have not the correct matching reference dataset. So it seems to be more robust. One reason for this could be that it also includes allele frequencies in the CNV estimation. We explored also other aspects in the benchmarking, such as how good can these methods identify the subclonal structures and how much resources do they require. If you are interested in these other aspects, please have a look at our poster or contact us directly. In the end, we can draw two main conclusions. All scRNA-seq callers were able to estimate the general CNV profile, given that the dataset was of high quality, such as having a good coverage. However, there's no single best method, but depending on the exact use case, different methods are best working. With this, I want to acknowledge my group, especially my supervisor, Maria Colomitache, and my colleagues who helped me in this project, Katja, Maria, and Dima. If you are interested into this, you can also look into our GitHub page where we show the SNAKEMAK pipeline for the evaluation and more results.